All right. Hi, everyone, and welcome. Happy Wednesday. Happy Gene Regulation Day. Always a big day for AP Bio, and for some reason, a very dreaded topic. But it shouldn't be because gene regulation is super important, and it also makes a whole lot of sense if you think about why genes and bacteria or humans or whatever it may be are doing what they're doing. It's all for a purpose. And so that's what we're going to investigate tonight. So, gene regulation. Okay, so first and foremost, we should talk about the purpose of this because that's how I've been starting out all of our little screencasts lately. But it's important to know before we start, or before we kind of dive in, like why we're even talking about this and why it's important in a biological sense. So the reason that we have gene regulation is to save energy, really. That's the main purpose, right? So we're only going to turn on certain genes at certain times. And that's just like a good practice for cells, especially bacteria who don't make very much energy, in order to make sure that they have, they're only spending energy on things that they need at the moment, and they're not spending energy on things that aren't currently necessary. The other thing to realize is not all genes are used at once, right? We're not like transcribing and translating every single one of our genes at the same time. That would be nuts. And also not all genes will be used by a cell in its lifetime. Like your skin cell is never going to produce the genes that your eye cell would produce, or your hair cells would produce, or your heart cells would produce, right? They all have the same DNA. Don't get that confused, right? Our skin cells, our heart cells, our brain cells all have the same DNA in them. They just are going to express different genes, the genes that are necessary for their role. And so gene regulation is a big part of just turning on the certain genes that we need for that function or at that time. All right, so anytime we talk about regulation, there are going to be many different examples of this, but it's all going to be based off of feedback, some sort of feedback, usually from the environment of the cell, that's going to tell the cell whether we should keep producing this thing or not. We're actually going to get into a lot more detail about this guy right here. This is the Trip operon that we'll talk about for tryptophan. But as we are moving through this pathway, you'll actually notice that our product, tryptophan, is going to come back up and actually inhibit the production of more tryptophan. And this happens really, really frequently. This is called negative feedback, right? So anytime that we have some sort of product or something that's coming back and inhibiting something prior, that we call negative feedback. And it happens in our body all the time. Like when we get too hot, we start to sweat and cool down. When we get too cold, we shiver and warm up. This is the same idea. When we make too much of something, or when we have enough of something, we're going to inhibit the production of that thing. And there's many different ways that we can regulate that. That's really what we're talking about on a large scale here, is how can we regulate and only turn on and off genes that we need when we need them. Okay. I'm going to spend a lot of time on this slide because there's a lot of important things that you need to know about here. But first, let's talk about the unit of genes. So we're going to start by talking about prokaryotic gene regulation. So prokaryotes regulate their genes, eukaryotes regulate their genes. Oh, I'm so sorry. Ooh, but they do so differently. Okay, so prokaryotes are going to be simpler, of course, but from an AP Bio standpoint, you actually need to know more about prokaryotic gene regulation. So even though it's simpler, you need to know more about it, as opposed to eukaryotes, which are more complicated, but you need to know less about for like the AP Bio test. Okay, so we're going to spend a good amount of time on this slide to make sure that you guys have a deep understanding of what an operon is and why we would regulate such a thing. Okay. All right, so an operon, this word up here. An operon is just a set of genes that have a similar function, okay? So like in this example, we're gonna be talking about something called the Lac Operon. And the Lac Operon makes 3 different proteins. So it's 3 genes that make 3 different proteins that are all involved with breaking down lactose. Okay, so lactose ends in O, so it's a sugar, and you've probably heard of it before. It's in milk, right? So all types of dairy, and it's a complex sugar. It's much more complex than glucose is, okay? And so we have 3 special enzymes from these 3 genes that are going to help us to break that down, okay? And all three of these genes are going to be involved in this process, so they're actually grouped together in this thing called an operon, okay? Kind of like how if you wanted to turn on all of the lights outside of your house, maybe you have one switch that does that turns all of your exterior lights on. You don't have to go to each individual light bulb outside of your house and turn it on individually. You have one single switch that will turn all of those lights on or turn all of those lights off. Okay, so this is the same idea. We are putting all three of these genes that do a similar function together, and we have one switch that's going to turn them all on or turn them all off, okay? And this helps us to regulate when we need to make something, okay? There's a couple of other regions that you need to be familiar with, and here we go. For whatever reason, it's not labeled on that picture, so I'm just gonna skip ahead really quick. This is the leftmost side of that picture, right, that you can see right here, okay? But there's a promoter region of the DNA, there's an operator region, and there's something called the regulatory gene, okay? So the promoter region is the region in which RNA polymerase binds, okay? Kind of like when we talked about DNA replication and DNA polymerase needed those little RNA primers in order to bind and start replication. The promoter is the special sequence of DNA in which RNA polymerase will bind. Right next to it is something called the operator, and this is going to be where our regulatory protein binds. Okay, so I'm going to flip back a slide really quick and talk a little bit about the regulatory gene. So this is the regulatory gene, and you'll notice it's actually not technically in the operon. It's not going to be turned off or on. It's just going to stay on, okay? And this regulatory gene is responsible for making a regulator that's going to help us regulate gene expression. Okay, so in this case, we have this regulator or regulatory gene that's being transcribed into mRNA and then translated into a protein that we call the repressor. Okay, and the repressor is going to actually fit into our operator region, as you can see right here. And when it does, it blocks transcription from occurring. Okay, so the end goal here is that we are able to make mRNA of these three genes, right, that then turn into three distinct proteins. But if we block RNA polymerase, right, which is the enzyme necessary for transcribing DNA, then we're not able to do that process, and we won't make any of those, we won't make any RNA, and therefore we won't make any proteins either, okay? So when this gene is off, it's because this repressor has bound to the operator and therefore blocked RNA polymerase from going through the process of transcription, and therefore also inhibiting translation and the creation of whatever proteins that set of genes codes for, okay? So as I kind of alluded to earlier, this is just a switch, right? And when the switch is, when the operon is on, it's because no repressor, nothing is blocking RNA polymerase from doing its job. It can freely transcribe these genes. And when it's off, it's because something is inhibiting or blocking RNA polymerase from doing its job. It's usually something called a repressor, okay? Okay, so that is that general premise. I'll go into a little bit more details, and then we're gonna do some comparing and some contrasting too. But those are the major regions that you need to be familiar with for an operon. So you need to be familiar with something called a repressor. You need to be familiar with operator and promoter. You need to be familiar with the genes that follow and why or polymerase would be able to go through those genes, okay? So I have all of that written down for you. So operon, entire stretch of DNA required for production of a certain protein. The operator is a segment of DNA where the repressor binds. It's the switch. The repressor is the protein that switches off a gene, right? So it binds to the operator and blocks RNA polymerase from moving. The regulatory gene is a gene upstream of the coding region that codes for the repressor protein. The co-repressor, which we haven't gotten to yet, but we will, is a molecule that binds to the repressor to activate it. And the promoter is the region in which RNA polymerase binds. Okay, so those are all our key vocabulary there. So, operons can be, now that you know all of the moving parts here, operons can be either repressible or inducible. Okay, this is all for negative control, which is going to be my main focus right now. And negative control means that we are using a product to inhibit or turn on this process. We're not amplifying a process, we're either turning off or on a process. So if an operon is considered repressible, okay, and remember an operon is a set of genes that all code for a set or for certain proteins. So we have a set of genes that we consider repressible. This means that the way that this is set up, this set of genes is capable of being repressed, okay? So the reason I have this spelled out so much is because if you can remember what these words actually mean, you'll be much more successful at remembering what these operons do. So if you remember that repressible means capable of being repressed, you know that repress means to stop or halt or turn off, then you will be able to remember that a repressible operon is capable of being turned off. That means that it's naturally or in its normal state or when nothing else is present, this operon is on. And by on, I mean active and producing protein, right? So RNA polymerase can move freely and make mRNA, which can then make protein, okay? This thing is able to be turned off if need be, right? But its normal state when nothing is present in its system is going to be on or active, okay? The example of this is something that we'll talk about in a little bit, but it's called the Trp operon, TRP operon, which is responsible for making tryptophan, which is one of our 20 amino acids, okay? All right. Operons can also be what we call inducible, which is capable of being induced or turned on. So to induce, I always think induce means like to induce labor would be to start labor or have labor occur. So if you're capable of being induced, that means you're capable of being started or turned on. So what this means is, so if you're capable of being started or turned on or activated, that means that normally when nothing else is around, this operon is actually off, okay? So it's only going to be turned on or induced in certain circumstances when something else is present that induces it, okay? But otherwise it will be normally off. An example there would be the Lac Operon, which we'll talk about in a second, which is responsible for making an enzyme to digest lactose. And this should make sense that we want to be able to turn it on, but that it's naturally going to be off because there's not always lactose in a, sorry guys, I'm in the middle of a podcast. You're good. Which means, sorry, some of my students just walked into my classroom. But we're only going to make the enzymes necessary for digesting lactose when lactose is present. Okay, so that's why this is going to be a good choice. And if any of that sounded confusing, I don't blame you. It is a relatively confusing topic. What I would ask that you do is just stay tuned for a little bit because I'm going to show you examples of this that will make more sense as to why we use these types of operons and what they are useful for. Okay, so an inducible operon has a regulatory gene, okay, that is going to naturally make mRNA that codes for a protein that is active. Okay, so the repressor that is made by the regulatory gene of an inducible operon is naturally active. Okay, and we can see that here and it's glowing to show us that it's active. Okay, what that means is in a normal state, when nothing else is around, remember these are prokaryotes. Specifically, these two operons actually both exist in E. coli, and E. coli lives in your intestinal tract. So we can talk about how what we eat actually affects these. But in a normal state with nothing else around in its system, this operon will produce an active repressor protein that can then come right in and fit into our operator. Okay, and when it does that, when it fits into the operator, remember, it prevents RNA polymerase from binding with the promoter correctly, and therefore RNA polymerase is not binding. We have I always think of it like a tree in the road. Here's our car, here's a tree in the road we can't get past, and therefore we're not making any RNA, which means we're not making any protein products, okay? I should also mention, so this is the lactose operon, the Lac Operon, right? And the products that we're trying to make are what we were talking about in the beginning of this podcast, which is enzymes to digest lactose. Okay, so in this instance, there's no lactose around, right? I said there's nothing else in the environment. So when there's no lactose around, we're not going to bother to make the enzymes to digest it. Okay, so I alluded to this earlier as well. Lactose is a very complex sugar. So we would much prefer to break down glucose as opposed to lactose. So glucose is just much easier to break down. It goes right into glycolysis, and we have a better situation with that. And so if we don't need to, we don't want to make enzymes to break down something more complex, because it's just a waste of energy. It's costly from an energy standpoint. Okay, so that's why we make this nice active repressor that fits right into the operator and prevents RNA polymerase from going through the process of transcription. And therefore, we're not making any of the enzymes necessary to break down lactose when lactose is not present. Okay, so if you haven't had any dairy in a while, then all the E. coli in your stomach currently have this situation going on in their operon, their Lac Operon, because there's no lactose for them to break down, and therefore there's no reason to make those proteins to break it down. Now, let's say you just drank a milkshake. Then there is tons of lactose, and the E. coli in your stomach will start to break them down. So here's what happens. That lovely active repressor, right, that was bound up here just a minute ago in our first example, actually is going to bind with something called allolactose, which is an isomer of lactose. Kind of always just think of it as like lactose itself, right? And so when you have a bunch of lactose in your system, this allolactose binds to the repressor, and you'll see it fits in very nicely, and it kind of mutates or bends the repressor protein to a point where it can no longer bind to the operator like it once could. So you'll see in this kind of oversimplified puzzle piece example, the arms of this puzzle piece are now sticking out, and therefore they can't bind to the operator because they're bound to an inducer molecule, is what we call it. An inducer molecule, which in this case is an isomer of lactose, okay? So when there's lots of lactose present, it's going to go ahead and bind to all of these repressors on all the operons, and the repressor is going to be bent in a way that does not allow it to bind, okay? Without the repressor in place, there's nothing blocking RNA polymerase from going through the process of transcription. And so RNA polymerase will do just that. It will travel down through the 3 genes. It will make an mRNA copy, which then in turn makes proteins. And those proteins are all they're all enzymes in this case, that are going to digest lactose. Okay, so they're going to get to work digesting all that lactose, breaking it down so that we can get all of the energy out of it. And they'll break it down, break it down, break it down, break it down, all the way until they've gotten to even the lactose molecules that are bound to the repressors. And they will break those down as well. And when they do that, we will change from this inactive repressor back to a active repressor, because it will no longer be bound to lactose. And we'll move back to this state where the active repressor can bind, and we won't make any more enzymes because we don't need them anymore. We've already digested all of the lactose, so there's no need for those enzymes anymore. Okay, so it's a really great energy-saving operon. If you drink milk or have cheese or any sort of dairy in your life very frequently, then the Lac Operons and the E. coli in your intestines are just going to be on pretty much all the time. And that's totally fine, right? But the moral of the story is we want to use an inducible operon in instances where we only want to make things when something else is present, right? So we use this with nutrients all the time. Like anytime we need to digest nutrients, the bacteria will pretty much use an inducible operon because they only want to produce the enzymes to break it down when that thing is there. Even if it's there quite frequently, doesn't matter. We still don't want to waste our energy making enzymes if it's not there at the time. Okay, so like if you go a few days without having lactose, for me, I don't know if that's even possible because I love cheese and I eat a yogurt every morning. So my E. coli are probably doing this quite frequently. But if that is not your situation, then yours would be off more often than others. Okay. The moral of the story here is that we're going to use this in times when we need to usually break something down or metabolize it only when that thing is present. And so then we use that thing or something like it as an inducer molecule in order to get this process started. I always tell my students, it's like, hey guys, it's lactose here. I'm here. Start breaking me down, right? It's like, hey, wake up, wake up. Let's get rid of this repressor. Let's get going. Let's get cracking. Break me down. That's kind of what this is signaling to our cell. Okay. All right. So that's an inducible operon. It's capable of being induced. It's capable of being turned on. And it's turned on in the presence of an inducer. On the flip side, we have something called a repressible operon. Okay, remember this is capable of being repressed. It's capable of being inactivated, which means when nothing else is present in the system, it is going to be active. Okay, so here's our regulatory gene again, but it's different this time. Okay, instead of making that nice, perfect repressor that we saw over here that fit perfectly into the operator the first time, now in a repressible operon, we're actually going to make an inactive repressor from this process. Okay, so you'll see up here. Remember, the regulatory gene is always on. Okay, it's not like it's inactivated, and that's why we have an inactive repressor. It's on. It just so happens to code for an inactive repressor, and you can see here that this repressor is not going to be able to fit into the operator. So when nothing else is present in its natural normal state, a repressible operon is on. Okay, and the RNA polymerase is moving down. There's actually 5 genes in the operon for a trip operon, and these genes are all going to code for proteins that are going to then combine. So you can see the polypeptide subunits. So they're all going to combine and make enzymes that are necessary for tryptophan synthesis. Okay, so tryptophan is an amino acid. It's not super common in diet. It's very common in turkey. I'm sure a lot of you have heard of tryptophan because of turkey, right? But it's not super common in other things. And so E. coli is actually capable of making its own tryptophan because we need amino acids always, right? Because we're always making proteins. And so we need amino acids in order to, make all those proteins, okay? So if we don't have tryptophan, we're in big trouble because when tRNA comes to drop off tryptophan, there's nothing attached to it and we have a big old hole in our protein. So this is a perfect candidate for a repressible operon because we need its product all the time, and repressible operons are naturally on. So we're going to be making tryptophan all the time, which is good because we need it all the time. You follow me, okay? So the only time we would turn this guy off is if there's either an overproduction of tryptophan or if there's a natural source of tryptophan. So for instance, if these E. coli are living in your intestines and you had turkey on Thanksgiving, right? Then the turkey you ate had tryptophan, and by the time it got to your intestines, your E. coli could stop producing all of these proteins because they had a natural source of tryptophan in their environment, and they could just use that instead, okay? So when we have enough tryptophan naturally, or we've made enough tryptophan to sustain us for a little while in making proteins, then we're not going to waste the energy to make a protein that makes more of something that we already have, right? That would be a waste. Okay, so here's what happens. Tryptophan is going to form something called a co-repressor, okay? That's going to help the repressor, like a co-captain, right? Co-repressor. And it serves as its own co-repressor to basically be like, hey guys, there's enough of me here. You can actually stop making more of me. We're good, okay? Or you know what? Miss Sheep just ate some turkey, so there's lots of tryptophan available. We can stop making some for a little bit and just use all the tryptophan from the turkey she ate, okay? And so it fits nicely into our co-repressor, and it makes it active, okay? And this looks more like the repressor that we saw from the inducible operon. That can fit very nicely into the operator, okay? And now RNA polymerase will not go through to transcribe or translate genes. Okay, because it is unable to do so due to the fact that the co-repressor is there and blocking its way. Okay, so I just want to summarize a few things. So with the inducible operon, it's naturally going to be off if nothing else is present. It will only turn on if an inducer inactivates the repressor so that RNA polymerase can come and make the genes that were coded for. Okay, and that will happen with lactose only when it's present, and that's going to be our most common example is the Lac Operon. So it's a good one to remember. This is also going to be used for things that we don't need all the time, right? Like enzymes to digest something that's not always in our environment, okay? On the flip side, the repressible operon has a regulatory gene that makes an inactive repressor. So this is in its natural state going to be on. Repressible operons are much more common for things that we need all the time, like tryptophan, and they will only turn off when a co-repressor, in our example, tryptophan, activates the repressor and therefore stops the production. Okay. All right. So that is operons in a nutshell, and that is the major way that prokaryotes regulate their genes. And again, this is all happening at the level of transcription because we're really just regulating whether or not RNA polymerase can freely move over the genes. Okay, so that's what's going on there. All right. Eukaryotic gene regulation, as I mentioned, is much more complex. You also need to know less about it. There are a few things that I will tell you about, but for the most part, as long as you know that eukaryotic gene regulation is much more complex, you'll be in pretty good shape. Most of the time we talk about prokaryotic gene regulation. Eukaryotes, though, a really important similarity that you should be aware of is that eukaryotes most commonly also regulate gene regulation at the level of transcription. Okay, so somewhere up here, we will have some sorts of regulation, and I will show you an example of that. Okay, or even somewhere up here with chromatin modification, I'm going to show you an example of that as well. But we're going to regulate most of the time one of these first processes, okay? And hopefully that makes sense because just like with prokaryotes, we don't want to make a product and waste all that energy and then be like, oh, we're actually going to have degradation of the protein, which means breaking down the protein, right? We don't want to make a product and then be like, never mind, JK. I know we just spent all that energy making this, but let's just like break it down because we actually don't need it right now, right? That doesn't make a whole lot of sense. That would be like if you were a factory owner and you were like, all right, guys, we're going to make a bunch of iPhones today. And then once you've made a bunch of iPhones, you're like, just kidding. We actually don't need all those. Let's destroy them, right? You wasted a lot of time, money. You wasted a lot of materials, right? It would be much smarter to just come in that day and be like, hey, guys, we don't need to make any iPhones today, right? So that's why we're mainly going to regulate these first few steps as opposed to something down here because we don't want to waste any energy making something that we're then not going to use, right? It would be like if you did like homework that like all of a sudden you're never going to use again and you're like, this is a useless waste of my time. I'm sure my students have been there before. I'm sure you guys have been there before too. Okay. So a couple things that I want to highlight. So this is just, I tried to make these a little bit bigger so that you could read some of the different steps in which we can regulate our own genes. Okay, so that's what's being shown there. Obviously, a lot more things can be done here than with prokaryotes, okay? But a couple of major ones to highlight. One is regulation of the chromatin structure. So something to know about DNA is that DNA is actually wrapped around these proteins called histones. Okay, and it's kind of like wrapping up a ball of yarn. Okay, so we actually wrap the DNA very tightly around these things called histones, and then these histones are all bundled together into our chromosomes, right? And what that does is it allows us to compact all of our DNA into our chromosome structures, which makes it a lot smaller than it would be if we just laid all the DNA out in a line. I have to double check myself, but I believe if you laid all of your DNA out in a line, you would have 2 feet of DNA in each cell, which obviously is impossible because your cells are like micrometers small. And so we wrap it around these little protein structures, kind of like we would wrap a spool of thread. Okay. Now, how we can kind of regulate what genes are being transcribed and translated is that we can do something called acetylation. Okay, and what that means is right under here, right over here to the left, we have unacetylated histones, and you'll see they're all bunched up. Okay, so picture like you wrap a bunch of yarn around a spool, okay, and you want to try to get to the yarn that's like at the bottom of the spool, right? But there's all this other yarn that's wrapped up around on top of it. You're not going to be able to get to that region, okay? That's what unacetylated histones would look like. We have something called acetylation, which can happen when we're ready to transcribe, which actually starts to unravel the chromatin so that we can transcribe the genes. And you can see that like now we have some loose fibers, some loose DNA that we can get at and it's exposed so we can actually transcribe and translate it. And so the process of when we acetylate and where in our body can help regulate what genes are being transcribed and what genes aren't, because if they're all wrapped up tightly here, we can't transcribe them. But here, since we've unraveled them a bit, we actually can go through the process of transcription, and that acetylation can be monitored so that only certain genes are being transcribed. So some other regions that are important, we have lots of enhancer regions, okay, and control elements that are actually way upstream, way earlier than our actual mRNA sequence, or sorry, DNA sequence. And so you can see here we have all of these different regions called enhancers. And basically, we have different proteins that have to come together in order for this gene to actually be transcribed. And it looks a little something like this. So we have activators, right, that will actually hop onto the enhancer, which will allow us with a DNA binding protein, okay, in order to get all of these mediators together with some transcription factors so that we can bring all of these things together in order for RNA polymerase to bind. You don't need to know the names of all of these. You just need to know that there's a lot of moving parts that come together when it's time to transcribe and translate various eukaryotic genes, right? So that we still have kind of like the repressor, like activators that are like, hey, it's time to go. Go time, this is the gene that we need. We have transcription factors that are coming in and mediator factors that are coming in order to make sure that RNA polymerase can bind. Similarly to how we had like a repressor that was inhibiting transcription from happening with prokaryotes, okay? With eukaryotes, we just have mainly activators and enhancers that are working to increase gene production when we need it in certain genes, in certain cells, at certain times, okay? Another thing that you might hear your teacher talk about is something called a TATA box, and that's just a bunch of TAs and TAs, so thymines and adenines, and that's going to be a region where the RNA polymerase knows where to bind, kind of like the promoter. Well, it's part of the promoter for eukaryotic cells. Okay. All right. So those are honestly the two main ones that I'm going to talk about. It doesn't really make a whole lot of sense to go into details about all of these other things because they don't show up on the AP exam. So it's not really worth your brain space. But I just wanted to highlight a few to show you how much more complex and how many more layers we have in our own DNA regulation as opposed to that of a prokaryote. Okay, we're actually going to hop back to prokaryotic gene regulation really quick because I want to show you a very interesting graph. And this was the long free response from 2016. So let me actually, 2016, I'm going to actually put the answer key in here for you guys because since we don't have anyone tuning in live now, it probably would be beneficial for you guys to work through this on your own and then look through the answer key. So this is question 2. So I guess I'll post the actual free response questions first so that you can pull it up without having to listen to me actually talk through the answers. You can pause me and actually take a look at the question 2 right there. And then when you're done with that, you can either press play again and listen to me explain the answer or look at the scoring guidelines that I'm about to post so that you have a good idea of what's going on here. So first of all, I guess you should feel lucky that you weren't taking the AP Bio test in 2016 because this is a very challenging question. It was one of their long free response, right, because it was question 2. It's worth 10 points. And most students, almost no students, actually realized that this was a gene regulation question, which is probably part of the reason why it didn't go too well for many people. It also has a really funky graph that confused a ton of students, okay? So I'll start by talking about that. We have a solid line in the middle that's labeled bacterial population density. And this solid line corresponds with the leftmost y-axis, which is also bacterial population density. And then we have a bunch of numbers here, right, ranging from 0 to 10 to the 9th that are representing that bacterial population density. And so we'll use the left side, the left y-axis, when we're looking at this black solid line in the middle, okay? But we will not use this axis at all when we are looking at both sets of dotted lines, because our dotted lines are nutrient concentrations. So we have nutrient 1 and we have nutrient 2, and then we have relative concentration of the nutrient on the right-hand axis, okay? And so a lot of students right off the bat were like, ooh, there's more bacteria with nutrient 2 than nutrient 1. And that's actually not true at all, because these heights that you see here on the y-axis are totally dependent on or totally related to nutrient concentration and actually have 0 to do with bacterial population density. Okay, so what we can look at is we can see right off the bat that nutrient 1 is being depleted first, okay? And that's leading to our first increase in bacterial population. And then we have nutrient 2 being depleted second, and that's leading to our second increase in bacterial population, okay? But it's a very confusing graph to read, so that led to a lot of problems for students, okay? There's a lot of questions that are going on here as well that we'll get through, okay? So question A is to estimate the maximum population density in cells per milliliter for the culture and describe what prevents further growth of the bacterial population in the culture. Okay, so I'm just going to touch on this question ever so briefly because it's not actually a question regarding gene regulation, okay? But maximum population density was a super easy one. You just had to look at the bacterial population and then look over at the left-hand axis, and it's at 10 to the 8th cells per milliliter that we reach a maximum, okay? So that was actually a point. Just from getting that, you get 1 point. The second part of this question, and remember that every bolded word is worth a point. So the second part of this question is using the data to describe what prevents further growth of the bacterial population in the culture, okay? So different things that could have been said here. Well, actually, it's really only one, and it's very simple, but a lot of students seem to miss this one as well. It's just noting that we stop growing when there's no nutrients, okay? So that's all that AP was looking for as well. We run out of nutrients, and so therefore growth stops, okay? The hint here was that it was simpler than you might think, is that you have to use the data here. So there has to be something on the graph that we're using to describe the preventing of further growth. And the only thing that's on this graph besides the actual population of bacteria to begin with is the nutrients. So we have to think, okay, we're not growing anymore because we ran out of food, right? And that's actually 2 points, right? And this question scored very low. So if you could get 3 points on this question, you would meet or surpass the national average, which means you're setting yourself up for a 3, right? That's what the national average is. Okay, part B was 1 point. Using the data, calculate the growth rate of the bacterial population between hours 2 and 4, okay? So if you don't know this already, a rate is always gonna be a slope. You could also see that because this is the units for the y-axis, and this is the unit for the x-axis. So this is y over x, right? Y divided by x, which is going to be slope. And we're looking between hours 2 and 4. So for this, you would have to pull out your calculator and realize that we're about at 10 to the 1st at hour 2. And then at hour 4, we're up to, it looks like 10 to the 4th, okay? So you would do 10 to the 4th minus 10 to the 1st over 2, and that would give you your answer here. And that's just simple slope formula, right? Y2 minus Y1 over X2 minus X1. And that should give you 4,995, okay? And that's going to be the rate there. So again, 2 hints there. The fact that it says rate should always tell you that you're dealing with a slope. But on top of that, the units given are indicative of the y-axis being divided by the x-axis between those 2 lines. Okay? All right. Neither of those are actually related to gene regulation, but parts C and D very much are. So part C is 3 points. Okay, first point comes from identifying the preferred nutrient source of the bacteria in the culture. Second point comes from using the graph to justify why you think that is the preferred nutrient source. Then the third point comes from proposing an advantage of the nutrient preference. Okay, so 90% of my students and 90% of students think that nutrient 2 is the preferred nutrient source because it's at a higher concentration, because some of them thought that, oh yeah, there's more bacteria there, even though that's not the axis that you use for nutrients, because it lasts longer, because here's what you actually need to think about. Okay, so this is how I walk my students through this question. Why is the nutrient source decreasing, right? Not because it's disappearing or disintegrating or something like that. It's decreasing because the bacteria are eating it, right? They're using that source for energy, okay? So nutrient 1 is being depleted first, followed by nutrient 2. You also need to realize that bacteria are not capable of knowing that they need to save the best for last, right? Bacteria don't know that you always need to save that bite of mac and cheese for last, because that's what's going to round out your meal perfectly, right? So they are going to use the nutrient that is most beneficial to them first, right? That is their preferred nutrient source first. And then they will use, if they still need nutrients, right? They will use a secondary nutrient source, okay? So your first answer here is that nutrient 1 is your preferred nutrient, okay? And again, the reason is the bacteria are using it up first, okay? It's always going to be the preferred nutrient source if they use it first. Why? I just said why. So the easiest way to explain this is that the bacteria are using it first. When both nutrients are present, only nutrient 1 is used, or nutrient 2 is only used after nutrient 1 is depleted. All of those are your possible answer choices there. But basically, you need to be like, hey, this is used first, so therefore it is the preferred nutrient source. Okay, so we got a point for mentioning nutrient 1. We got a point for saying it is the preferred nutrient because it's used up first, okay, or it's used up before nutrient 2. Okay. All right, the last part of this question is to propose an advantage of the nutrient preference for an individual bacteria, okay? So basically this question is saying, why would a bacteria have a preferred nutrient? Why do we use nutrient 1 before nutrient 2? Like what's the big takeaway here, okay? Why would that be important? And the bottom line is it's the same reason that we regulate genes anywhere. It's gonna be because of energy, okay? So you need to think of a reason, and there's not a specific answer here that's given or anything like that. You just need to logically think like a scientist. Okay, what is nutrient 1 doing for our bacteria that makes it a better nutrient source than nutrient 2? Why would they favor that? Okay, there's a couple different answers that you could give. So one, as I just said, we're saving energy, like we're not using energy when we don't need to, okay? Or we're not using energy to make all of the enzymes to break down both nutrient 1 and nutrient 2 when we can just focus on one at a time. Perhaps the nutrient provides more energy. Perhaps nutrient 1 is more energy rich than nutrient 2. Do we know that's true? No. Could it be true? Sure, right? You're thinking logically like a scientist, and so you'll be rewarded for that. And then another one would just be that we don't have to express all of our genes at once, okay, because that's too energy costly for the cell. And that's why we have operons in the first place, okay? So this question, this proposed advantage is like, hey, why do bacterial cells have operons, right? And so that's your first two answer choices if you're following along on the rubric. Does not spend energy making enzymes or proteins the cell doesn't need, or does not have to express all the genes at once. Then that third option is just logical thinking, right? Maybe that nutrient is actually more energy rich, right? Like glucose is going to be more energy rich than lactose, right? So if this was a glucose or lactose situation, then this would be glucose being used up first, and we wouldn't start breaking down lactose until after we had run out of glucose, right? All right. Our last part here. So far we've earned 1, 2, 3, 4, 5, 6 points. So 4 points remain for part D. We're going to describe how nutrient 1 most likely regulates the genes for the metabolism of nutrient 1 and the genes for the metabolism of nutrient 2. So that's 2 points because there's an and there. And then we're going to provide 2 reasons, so also 2 points, that the population does not grow between hours 5 and 6. Okay. So for part D, this is a straight up Operon question, but it's also a logical thinking question. So they're basically telling you, okay, nutrient 1 regulates the genes for its own metabolism, for its own breakdown. And nutrient 1 also regulates the genes for the breakdown of nutrient 2. How does it do that? Okay. You don't have to have a super complex answer here, but you need to think back to operons. Okay. And the answer that they're looking for basically is that nutrient 1 is going to enhance or promote the metabolism of its own enzymes, right? It's going to be like, hey guys, I'm here, kind of like lactose, right? Hey guys, I'm here, I need to be broken down, okay? So it's gonna promote the genes for its own metabolism, and then it's going to inhibit or repress the genes for the metabolism of nutrient 2. That's all they're looking for here. You need to be like, okay, it's breaking down itself first, so it must enhance production of genes for itself, right? And it must inhibit the production of genes to break down nutrient 2 because it tells you that it regulates it, and otherwise we would be breaking down nutrient 2, right? So it must be putting a stop to that. So there's 2 points just for thinking logically there about enhancing one and repressing the other, okay? The last part talks about why we don't have growth between hours 5 and 6, okay? And there's a couple things that you can say here. So obviously between hours of 5 and 6, nutrient 1 has run out, but we are yet to break down nutrient 2, okay? And so reasons here, you could say that nutrient 1 is depleted, right? We've ran out of nutrient 1. Nutrient 1 is gone. We've used up nutrient 1, anything like that. Or you could say neither nutrient is being consumed at this time. You can also get a point for saying that, right? Because nutrient 1's gone and nutrient 2 is staying constant. We're not consuming any nutrients, therefore we don't see growth at this time, okay? Another reason you could say is that it takes time to produce the enzymes necessary to break down nutrient 2, right? So this is our transition phase. We've ran out of nutrient 1, so we're no longer repressing, right? Because remember, nutrient 1 was repressing the genes to metabolize nutrient 2. So we're no longer repressing the genes to metabolize nutrient 2, but we're going to have a bit of a transition phase as we start making those enzymes in order to break it down. So anything like that would get you the second point for reasoning. Those are really tricky points to get, and they actually, usually with questions like that, there's like a lot more things that you could have written to get points. And with this one, it's pretty small range. But again, and I would reiterate this a million bajillion times, with these questions, no one gets, I mean, a few people, but no one gets perfect scores. And that's not what we're shooting for, right? If the national average for this question was a 3 out of 10, then a 3 out of 10 would set you up, as long as you do well and do the same on your multiple choice and on all the other free response, to get that 3, right? If you're shooting for that 5, then maybe you should shoot for like a 6 out of 10 on this question in order to set yourself up for success there, right? But that still means you could have missed 4 points. Right? And so you kind of have to level with yourself. And this is also really good preparation for the test. You can't panic when you're like, I'm not sure, right? It is okay, but you should be able to write down everything that you do know in order to get the most possible points. Okay, but it's going to be really, really, really hard to get full points on any of these free responses. You're really not intended to do that, right? It's just to see how many points you can get and what you do know, okay? All right, the last thing is there's a button here to sign up for a free Kahoot session on the 19th of December. It's going to be our last session before the holiday break. It's free. It's going to be really fun. It's going to be like a huge game. It should be an absolute blast. So bring your friends, even if they're not members of Fiveable, and they'll be able to play too. And it'll be really good review before you go off into winter break and take finals, okay? That's it for tonight. Hopefully you understand gene regulation a little bit better now. And I will see you next week to talk about mitosis and meiosis. See you then.